that go from Spokane Community College and some people from Kellogg. And who else knows where people are from, but I'm pleased that you're all here. I'm Curt Nelson. I'd like to remind you of the special session today on science. This last century has three main themes that we've tried to portray. One is the development of the atomic bomb. The next one was the landing on our moon. And the third main thing that happened in the last century is the development of the structure of the DNA molecule. And I'm very pleased this format's a little unusual tonight, being a radio station kind of a setup. And we're very pleasantly surprised that we are going to try something new. This is a radio station, KNIC, right here. It's somewhat old and somewhat modern. And these gentlemen here, I'll let Rhena Cooper, who's going to be the interviewer rather, these famous personages over here. And at the end of the session, as usual, we'll field questions from the floor. So welcome aboard, and we're certainly glad to have you here this evening.
Welcome to KNIC Radio. We are live and on the air with guests Watson and Crick. And fortunately this evening we also have some in-house guests. Tonight our topic is DNA, the double helix, the code of life, the essence of our inheritance. These common phrases of today permeate our lives and our classrooms. They sound so normal and ageless, but in reality, the structural understanding of DNA is less than 50 years young. This is Rhena Cooper with KNIC Radio, and I'm thrilled to bring to you a conversation with Dr. James Watson and Dr. Francis Crick. These gentlemen are two of the key players in elucidating the complementary double helix structural model for DNA and receiving the Nobel Prize in 1962. Dr. Watson and Dr. Crick, you are so connected with the DNA story of this century. I've just got to ask, were you a child prodigy? Did you own a science kit? What drew you into the sciences to begin with?
No, I had a pretty normal upbringing. I attended the local grammar school in Northampton. That's where I was born. And I didn't have a science kit. I did have a nice set of Mies children's encyclopedias. They encompassed geography, history, art. But I was fascinated with the science part of those encyclopedias. I tried a few experiments. I tried to make silk and failed. And, I got into some explosives, which my father didn't appreciate. We came to an understanding of how to control those. But in reading those encyclopedias and thinking about planets and atoms and almost anything to do with science, it fascinated me. When I was 11 or 12, I went to my mother and I told her my worries that there's so many things being discovered and known already about science that by the time I got old enough to work in the field, that there'd be nothing left. And she assured me that there would still be a few things for me left to look at. My family wasn't scientific. My father made shoes. He and my uncle owned a shoe boot building company, which was very popular in Northampton. In fact, our mascot of the school system was the cobblers. So, my mother was a teacher, but she was not a science teacher. She was not specifically a careful thinker, but she was a good influence on me. So I would say I was a prodigy.
Great, and nice to know about the shoes too, and that there's still things left to discover. Dr. Watson, what about you?
I guess you could probably say that I was a prodigy. My real interest in science and just what basically life came from avid interest in bird watching, starting at a young age. And then it really translated into me finishing and going into college level work at age 15. So I entered college at 15, finished at 19 with a degree in zoology, and then went to pursue graduate work, and I was actually turned down by Caltech and Harvard. And then what I ended up doing was doing a PhD work in genetics, and that's what really turned me on to this whole idea of the search for whatever the gene was. And I finished a PhD at age 22. So I was a prodigy sort of in the later part of things as far as my educational experience, but I can trace it really back to bird watching.
Have the former schools regretted their turning you down?
I suspect they did. Caltech was where Pauline was at, and there was a little bit of jealousy going on there. And one of the things that happened, the director of the Cavendish lab that we were working under at the time that we discovered DNA, he was very much pleased that some of his techniques and so forth helped us beat out Caltech.
So Dr. Watson, you were a 24-year-old American, and Dr. Crick, you were a 37-year-old Englishman. How did the two of you end up working together at the Cavendish lab?
Well, after World War II, I had been serving as a civilian scientist in the weapons design, and the war ended, and I was offered a permanent job working in the scientific intelligence, but I knew that for a career I didn't want to design weapons, so I kind of took stock of my qualifications. I had a bachelor in science and physics from University College London. I had a degree in physics. It was rudimentary at best. I had no published papers. All I really did is I had an interest in that border between what was life and what we call non-living. And so I went to one of my mentors that I worked under during the war, Massey, and kind of proposed my problem to him. And I asked him, I said, do you think I would be any good at research? And he said, he kind of paused for a moment and looked across his desk and he said, well, you know, Crick, he says, people dumber than you have made it. That wasn't the encouragement I was looking for, but it was enough for me. And so we took that opportunity. He walked me over to Strangeways lab and introduced me to Maurice Wilkins, who ends up being a very important person in this process. And, I had interviews with physicists and chemists, and I wasn't so much interested anymore in physics. But there was a new field starting to open up. We still hadn't gave the title of biochemistry yet, but that did interest me, and so they offered me a project for a couple of years. I was looking at the physical properties of the cytoplasm, doing some cellular work, and it wasn't, very interesting in the project I was doing, but I did get some published papers, and got the opportunity to give some seminars to the people who worked there, and I just kind of started to form myself as a scientist. And I went to announce my progress after a couple of years to the head of the Strangeway Labs, and he had on his desk a new position at what they call the Medical Research Council. And it was working with the structure of proteins using X-ray crystallography at the Cavendish lab, and I told him of my interest in it, and he allowed me to fill out an application, and that's how I ended up at Cavendish.
Excellent. And Dr. Watson, what about you?
Well, I think you could trace it back to my experience in my PhD program at Indiana University. My graduate advisor was Salvador Doria, and he was instrumental, along with Max Delbrick, setting up the phage group at Cold Springs Harbor. In 1948, just prior to finishing my PhD, I went there and I took a course. There was a 3-week course that was taught at Cold Springs, and it was basically all of the techniques that Delbrick and Luria had learned about how to work with bacteriophages. And that was really sort of focused my interest on DNA, and what happened was my PhD dealt with bacteriophages and such. And following that, I just had an interest in trying to figure out what exactly this gene was. And a lot of the work at that time was focused around proteins. People had a lot of interest in that being the genetic material. We actually knew DNA for quite a period of time that it existed. We knew what it consisted of, the basic units of it. And it was far simpler than proteins. So one of the things that people hung on to for the longest time was the fact that we had the protein was the genetic material, not this thing that was found in the nucleus, described as DNA. We didn't know the structure of it. And as a result of that, many people followed the protein path. I chose to be different. I chose the DNA path. And one of the things that I had happened to attend a conference doing some postdoctoral work, and Wilkins had an X-ray image of DNA. And the thing that I saw there was just this ghostly image, and I couldn't get that out of my mind. And I was aware of that people were talking about this might be the stuff that genes are made of. And that led me to believe that somehow this X-ray diffraction technique was going to be critical in solving this problem. And I knew that the Cavendish labs were instrumental in developing that technique. And the director of the lab was the founder, basically, of X-ray crystallography. So that's the reason I wanted to pursue or end up at Cavendish, was to go into that aspect of it, because I thought that really would be the secret to discovering it.
How was it for you as an American in England?
It was difficult, and I think both of us have sort of different impressions on the discovery process. My perspective on the discovery of DNA, I know, is quite different from Crick's, and it's part of it's due to the fact there's an English fair sense of play that has to take place. They seem to be very much into cooperating with one another. I was probably more of a competitive sort. And we were sort of in a race. I felt it as being more of a race with Pauling. Pauling had described the alpha helix structure of proteins, and he was putting forth information. We were actually getting early manuscripts from his son, Peter, and as soon as they'd come, we'd pore over those, see what we could glean from those. So there was a sense of urgency on my part, but I didn't really perceive that from Crick's perspective. So I think there's a bit of difference there from the American competitive attitude, where it's more proper to sort of cooperate with one another, not be so competitive in the English sense. And one of the things that I was noted for, and I didn't impress too many people, but I grew my hair out while I was in Europe. I didn't want to be recognized as some of the personnel from the American Armed Forces, and there were several people that I upset quite a bit by doing that.
How long did it get?
Well, and it was just long on the ends of things and a little bit unruly when the wind was blowing.
Oh, okay.
When he showed up, he wasn't told to come visit with me because they knew I was interested in the problem also. And I wasn't home, but my wife was. And I got home and she said there was a bald American looking for you. And he wasn't bald, he had a crew cut.
Oh, okay.
But the hairstyle at the time was collar length or over. And it was just unique that when he showed up, he had the crew cut. But he quickly remedied that.
So when you guys were working in the lab, today we hear a lot about collaborative learning, team players, things of that nature. Were you guys just working on a project together? Were you friends? What made this synergy work for the two of you?
Well, odd enough, neither one of us were specifically directed to work on the DNA project. We both had our own arenas. I was working on the structure of proteins and trying to figure out the X-ray crystallography problem of that structure. But we became fast friends. We had a lot of things in common, our interests. We were friends not only in the lab, but outside the lab. We would spend time in the park. We just got along well. We talked well together. And what made us, I think, work, one reason we worked well together was that when one of us had an idea, the other was free to scrutinize it without causing a defensive situation between us. And that saved us quite a bit. There was a lot of, I guess you could say, scientific dogmas at the time. People were pretty convinced from Levin's paper that DNA couldn't be the genetic code. It was too simple. He announced that in his hypothesis. It was just too simple to code for something as precise and intricate as a protein. So it couldn't be the gene. But we were willing together in kind of our closed room to sit and discuss topics like that. And if we kind of got in a cul-de-sac, the other one was responsible for bringing us back out of it and saying this, this is untenable. Let's move on and look in this direction. So it was once said that we had to have it rocking the boat because a lot of people would say, Watson, quickly back to what you're supposed to be doing. And I don't know about Jim, but I seem to have my nose in what everybody was doing.
You were visiting other people's labs and.
Yes.
It was interesting because what would happen in Cavendish was quite a few research projects that were going on. He was notorious for waltzing into someone's lab and offering his theory about what was wrong with what they were doing. And then generally what would happen is he'd come back a day or two later and find out that his theory was wrong in and of itself. And there was a lot of people, especially the younger people that hadn't built reputations yet, actually feared him. So they tended to avoid him, and he wasn't too well liked by the director of the academy as well.
A common phrase that you hear often is great scientists stand and build their work on the shoulders of other great scientists. Who were some of the other key players in this DNA story? You've mentioned Pauline and a few of the others, Wilkins. Kind of tell us a little bit about what each of them contributed and where we were at this point.
Well, Jim had discussed a little bit about finding the structure of the alpha helix. So what that really did is it put helices on the mind of people working in the field. Before this time, it was just considered to be a straight structure, not to take on this helical formation. And so, as soon as he got done describing that, the secondary structure of the protein, he started working on this DNA question. And I happened to be working with Rosalind Franklin, who was over at King's College, who worked with Wilkins, although he never would admit that they were kind of working together. He always considered that she was somewhat his subordinate. But she was partially my research partner also. And they were doing the X-ray crystallography work. They showed a lot of the pictures that they got from doing this X-ray diffraction. They were instrumental in applying some of Bragg's equations and deciphering structure of DNA. But they were specifically working on the structure of DNA. So we were just, I shouldn't speak for Jim, I was just kind of meddling in their business. And anytime they had something of note, I would try to make my way over on the train and sneak a peek at it. Get a look in their files and see what it was to tell us. And I would come back and meet with Jim in the back room. They ended up putting us in a back room off the lab when it became open because the other people working in there got annoyed by us talking all the time. I think they were more annoyed that I kept sticking again my nose in their business, so to speak. I was told one time that I had a problem in that if I could even grasp the concept, I thought I knew everything about it. It frustrated a lot of people. So that's how Maurice and Rosie were instrumental also.
So when you were working on this and pulling together the pieces that you knew from the other stories going on and the helical structure, etc., when did you guys figure out that you were really onto something big and not just thinking you were onto something big?
I'm not sure where, because we had so many, there was a number of sort of false attempts that we were working at. And we had the information from a variety of people that we tried to bring together and piece the puzzle together a bit. And we were trying various things, and then we'd bounce that off other people, and they would suggest, no, that wasn't quite correct. You have to kind of go back to the drawing board. We made several attempts, and then we'd find out, look at it further, again, we were incorrect. But then what happened was we were waiting for some metal models to come back from the shop of atoms so we could put this together. And we were basically mimicking Pauline's way that he discovered the alpha helix. And what he did is he took basic principles of chemistry, how atoms would like to be next to one another, which atoms would be involved in doing that, and then also utilizing a model building approach to solving these things. So we used Pauline's approach that he used to solve the alpha helix, along with this X-ray diffraction data that Wilkins and Franklin was providing. And then one of the things that we ended up doing while we waited for these metal models to come back from the shop, I started taking some cardboard cutouts and lining things up. And there was sort of an intuitive event there that we lined them up in this one particular pattern, and it seemed to work. And I quickly called him in and had him take a look at it. And there was that moment of looking at it saying, that looks right. But we weren't sure of ourselves until we actually had the models built from that. Until we had actually the metal components that we could actually put the model together.
What was different about your model opposed to what everybody else was putting out on the table at the time?
Well, a lot of what was happening is people were looking at it, and we knew one of the things that we suspected is there was a regular structure associated with the sugar and phosphate in the molecule. And then there was 4 components that were associated that were different. And those components, the sugar phosphates, suggested some sort of regular pattern. And our several incorrect temps put those sugars and phosphates towards the center of the molecule, and the bases were sticking out. And we had to come up with all sorts of strange ways that would work chemically. And none of those seemed to work. And then one of the things that happened after while we were still working on that particular one, we got a report that Pauling was putting forth a model as well. And it was a 3-chain model such as ours, and it had the same type of setup, but he had a sort of strange twist on things that actually we looked at it and knew right away after reading it that his model, there was no way it could be correct. It was simple chemistry that he didn't seem to think of in putting forth this model. And it was rather interesting because to see that this individual that we looked up to really, and from his publications and previous work that he'd done, and then seemed that he'd make this sort of elementary mistake like such. But at first we thought he was possibly doing something very close to what we had. It was actually a little bit different.
There was about really 2 classic errors going on in this deciphering this structure. And the first one was that this sugar phosphate backbone was being put in the center of the model. And the reason for that was the other error that everybody assumed that the bases had to be exposed if they were going to be copied. And to turn the bases inward would mean that they would no longer be exposed. And this again goes back to the original thought that DNA at best was a kind of a scaffolding at which the protein gene would be built on. Not that the DNA coded for a protein, but the gene itself was a protein. And that it was linked to the DNA, and then it was able to hold the code. So the people who were trying to build these models were branching from that and making these mistakes and putting the structure together.
What was the attraction for so many people to hold on to protein as the genetic code instead of going to DNA as the genetic code? What would be an advantage to that?
Well, there's more basic units. There's 20 amino acids that build proteins in living systems. Where DNA, there was only these 4 bases that were different. And then the complexity of the code, you could make more complex code with 20 different units versus 4 different units. So people really were embracing that. And I think a large part of it potentially was due to ignorance. We didn't know what DNA specifically was, how it was hooked together. Where we were starting to find more and more information, there was a lot of information emerging in that protein. So people really were still holding on to that notion.
And they were also, looking at function and trying to decipher structure from function. They knew proteins were complex, so what coded for it must be complex. And again, it kept coming back that DNA is too simple. There's only 4 bases in DNA. How could you take something so simple and code for something so complex? So everything kept pulling back to that. And how would you make this structure with only 4 bases complex enough to do this? And that's where our model became different as we started working with it.
So what all did you actually put in your first paper, knowing there were other people out there working on these models?
When we did get the metal pieces together and we sat down and started to build it after the cardboard cutouts, one structure in the DNA is just where the 2 bases come together in the middle. And we sat and it took us about 24 hours to build a complete small model, not the one you see in pictures, but a very small model. And we had half of 1 base pair constructed. And we knew we had it. It just seemed to go together the way we were putting it. And I was excited. I knew we'd struck it, And while everybody was trying to work on it, we had finally figured this out. And
We waltzed across the street, the Eagle Pub in Cambridge, and he waltzed in and proclaimed that we discovered the secret of life. It made me a little bit uneasy when he did that.
I went home and I told my wife, I said, we've done it, we figured it out, we've made a discovery. And she told me later that she just kind of ignored what I said because I was always coming home saying that. I discovered something great. So when we sat to write our first paper on a structure, which we basically limited the paper to was the structure of DNA, I kept prodding Jim to bring in its genetic implications and and how this could be the hereditary material. And he was afraid. We only actually put one line in there because we did not want to be beat to the punch on announcing it as genetic material. But we did mention that, from the model that we postulated, it's recognizable that it's able to be copied. It could be the genetic material. But that was the only line that we put in there other than the 900 words on the structure of it. Jim was a bit afraid that we'd made a mistake and we were going to make kind of goose of ourselves. We wrote up a whole paper and we'd made an error in constructing this. A simple error like we were recognizing that other people were making.
But this didn't bother you?
Oh, not a bit.
I understand that your first paper was very much the structure, and you kept the implications pretty much to a minimum and stayed with the pure empirical of what you were doing. But your second paper came out really quick. Usually there's a time frame in between there. What all was happening that this would just amplify so quickly for what you guys were doing?
Well, again, I think largely the second paper would have been done in a manner as our first paper if we wouldn't have felt the rush to be followed. So the structural paper was more important to get that information out initially, just so we could sort of stake claim to this. And again, that's from my more competitive edge, I think, than from his edge. But if we could get that put forth and then just make this statement about the fact that this mechanism that we have as far as the structure would allow for a mechanism to copy the information contained within it, then we could follow that up. But then sort of wanting to get that information out as soon as we can. And again, I think largely is due to the competitive spirit, especially from my side.
Also, just briefly after we had put out that first paper and had already started to work on the second paper, Jim took a train ride over to King's College where Wilkins and Rosie were working, and I'd already explained how the relationship between the two was. Rosie wanted to work on one type of procedure for taking the pictures. There's a wet and a dry form of taking these pictures, and she got some really excellent images of DNA. And Jim went down there, and Wilkins had kind of snuck a copy of one of the pictures and showed it to Jim. And by looking at that, he was able to understand that what we were applying and discussing was true. He kind of calmed his fears that we were making a mistake. And so we went ahead and published the paper, put in the genetic implications. Again, be calling to the touch.
So how quickly in your own minds what kind of applications did you start thinking about off of this empirical research?
Well, I think you can, once you understand that information, and actually before we even can solve the structure, I have been doing some reading on DNA, what we've known up to that point. And one of the things that had become sort of clear to me is the fact that there was a number of experiments people were doing suggesting DNA was highly stable. And that worked well with me as far as looking at the idea that the gene would be something that would be highly stable material present within the cell and organisms for it to be maintained long term through generations. So that smelled right to me, that idea that DNA being stable. And I wrote out a basic framework where DNA then would continue that information and act as a template for RNA. I've done some work with a virus that had RNA present within it. And then that RNA then would be translated into a protein. So I had some early inclinations that what we now know as the central dogma was present from some of the reading that I had done. And now we can look at that and verify that that's what happens as far as the flow of genetic information. And then once that was determined, that's what we really saw a whole host of ideas can come forth from that. Once you make the leap from what is the genetic material structurally, then how does it function? And there was actually much work was completed by the people that were trained in the Flash group at Cold Springs Harbor. So much of those accomplishments that we do now in modern day DNA work was associated with that group.
This understanding of DNA and the flow of information and how it goes on and how it's used to make proteins, kind of led into some other big projects. Bring us up to speed a little bit on the Human Genome Project.
So Jim has been working on the Human Genome Project, and as of this month, March of 2000, there's only one partial chromosome left to be coded for. They've deciphered 22, they just finished the 22nd one. So I can tell you about his work there.
What my involvement with the Human Genome Project, I've been criticized for deeply concerning that, because I was asked to actually head this project up in 1988 until 1992. I was the first director of the Human Genome Project. It was largely due to the fact that I was quite successful at Cold Spring Harbor raising money. So I became more of an administrative person, largely after my affiliation with DNA, and I spent time at Harvard for doing some teaching in the biology department. But I became really intimately involved with Cold Spring Harbor in raising money and developing a real world-class research institute there. And again, many of these DNA spin-offs came from that group. And then in 1988, I was asked by the NIH to head up this project here, the Human Genome Project, where basically what we're doing is we're trying to go through the entire set of chromosome content we find in a human being and sequence that up. We want to know what the sequence of the 4 bases in DNA, the TDCs and Gs that are found within that. And that project was initiated in the early 1990s, and as Francis said, we're just about ready to have that project finished and complete. Just as far as what we know what the sequence is. One of the things it's going to take many, many years to figure out what does all that information mean. So just getting to the pieces of information is much like the DNA discovery. You can put that information together and know this is what it is, but what does that mean to us? So we're sort of at another threshold now where we have this large database of information. Now what does that information mean? Undoubtedly we're going to find gene locations in various portions of those chromosomes. Some are going to have medical spin-offs and so forth from that.
What, if we look towards the next century, your research, what type of problems out there on the horizon do you think we can look to this particular area to find solutions?
Well, we both agree on the fact that there's an overpopulation problem. How are we going to deal with the rise in the number of people on the planet? How are we going to feed them? How are we going to supply medicines for them? And there's quite a bit of research that will take place on making crops more productive. There's about 30 crops that feed 90% of the population. We need more lands to grow these crops on. Maybe we can engineer crops that can grow on poorer soils, grow with less water, less fertilizer, less pesticide application. There's also, as Jim was saying, the medical aspect. We can take genes required to make particular proteins that defend us against different types of biological invasions. And if you happen to be lacking that, we have to take a supplement. We can have a plant produce that instead of trying to do it with bacteria in a lab. Large proteins, you can't have bacteria synthesize the larger proteins that we use in medicine like you can with insulin. So possibly to use a larger organism to do human work will be a benefit.
Are you currently working in this area, or has your personal research moved another way?
I only stayed on a couple of years after this work was done at Cavendish. Jim and I sat and we worked with more of the deciphering of implications, how to follow this line from DNA to RNA to proteins. We worked in correspondence with some gamma from America on the 20 sites where these amino acids would be coded for. But I had another interest in neurobiology, so I taught there at Cambridge for about 20 years, and then I moved over to the United States, and I'm working down in La Jolla, California now at the Salk Institute on brain research, looking into dreams and vision and consciousness.
Watson, what do you see out there on the horizon?
But my focus with the Human Genome Project was largely disease. And if we could, and my whole premise from this was if we could somehow understand life better from the genetic level of detail that we now have the capability of figuring out, then we could end up having some better quality of life, and especially from the disease aspect of it. And if you can look at genetic disease, how that impacts people, if there's ways that we can start looking at those genes, understanding what's causing the change, what's the problem with the protein, it's not being coded for, it's being incorrectly coded for. If we can somehow start coming up with alternative ways to treat those people, rather than treating them symptomatically, actually get at the genes themselves.
Are you still in this area of research?
Well, actually what happened was I was investigated as being director of the Human Genome Project. I was investigated for having some potential conflicts of interest because of having investments in several biotech companies that could potentially benefit from this information. So I resigned from the directorship, and what I've done now is I've actually gone back to Cold Spring Harbor, and I'm acting now as the president. I'm no longer the director of Cold Spring Harbor Labs, but actually the president.
Is there anything in this story that you guys feel that we need to bring out, any pieces that we've missed?
Well, I have read many pieces and parts of interviews that we have given over time, and we're given a lot of credit for the discovery of DNA. I think it's important to understand that through the people that were working on it at the time, if we would not have figured the structure out, it was quickly to be figured. Maybe not within that week or the month, but there were enough people working on it that were getting close that what we were giving credit for was going to be discovered anyways. But what we did that I think we should be given credit for is that we picked a problem and identified it, that we were both very interested in. And the second thing is we stuck with the problem. If we needed more education on particular parts, like, to this day, my knowledge of chemistry, organic and inorganic, is minimal. But we would go learn about that part. We'd ask the right people, ask the right questions, and bring that back and apply this to it. So we stuck with the problem. And I think that I read one time, again, how important we were to this discovery of DNA, but I don't think Watson and Crick made DNA, but I do think DNA made Watson and Crick.
It's a very humble perspective for such a pivotal thing in our century. Any closing comments?
I would just echo his comments. Without the work of Franklin, without the work of Wilkins, without Greg's leadership in the Cavendish Labs, without Pauline's background material that we based our approach on doing things, I don't think we would have gotten to where we did. And there's countless other people where we made mistakes along the way that steered us in the right path. But I think that's true of a lot of scientific discoveries. Unfortunately, we remember one or two names associated with the discovery, not realizing the enormous amount of contributions that other people made up to that point.
So in the 1800s, we knew DNA existed. In 1953, these gentlemen, Watson and Crick, postulated a complementary base pairing model, but not just a model, an answer to how DNA replicates itself from generation to generation, an answer to how DNA is used to make RNA for the work of protein synthesis, a method of moving genes into bacteria to synthesize things such as human insulin for the treatment of diabetes, an insight into who we are and to how we are connected to the great web of life. A great century indeed. Not only have we reached out into outer space, we've gone into the nucleus of the atom, but we've gained a view of ourselves as we hang on the twisted ladder of life, DNA. At this point, thank you, Watson and Crick. At this point, we would like to ask Dr. Oppenheimer to come and join us up here at the front. And do you see some parallels between the people involved in the story of the atom bomb and the people involved in the DNA story?
Not particularly. I have the greatest admiration, of course, for what these scientists have done, especially for their humility if what you say is true, that DNA would have been discovered and its importance realized by other scientists. The same thing, of course, is true of the atomic bomb. If I had never lived, the atomic bomb would surely still have been built. The one at Trinity might not have worked. There's always the question of who brings the mastery to the application of science at a certain time. But what we know from the history of science is that discoveries are partly the agency of interesting men, and they're partly the agency of the times in which we live. Discoveries, interestingly, coincide with the time when the world was able to absorb them. And so I don't see any particular parallels between what you have done and what I did, but I do see a parallel between what you've done here and the periodic chart. It's all part of the Enlightenment's work to map the world. Lewis and Clark mapped the West. Thomas Jefferson gave us a rectangular survey grid system. You mapped the genetic structure of proteins. This is all part of an attempt to classify the world in order to make use of it. And the classical application of that of the 20th century was the building of the atomic bomb. But what strikes me as different, and I would be eager for your comments on this, what strikes me as fundamentally different about what I did from what you have done, is that we were working from quantum mechanics, and the more you know about quantum mechanics, the more puzzled you become, and the less certain the universe is, and the more one feels at sea in a world of physics knocking on the door of mysticism. I don't see anything mystical about what you've done. Ingenious, yes, but not paradoxical or mystical. Perhaps you could reflect a little bit about that.
Well, one of the things that we struggled with for a period of time is the X-ray diffraction data that we were looking at was suggesting either 2, 3, or 4 chains were involved in the molecule. And Francis insisted on working on 3-chain models. And I took the simplistic approach. So I think it is very much different from quantum mechanics. Because what we ended up doing, or what I ended up doing, sort of against his advice, was starting with the 2-chain model. And I wanted to rule out all the simple approaches. Why approach something from a complex point of view if it is indeed simplistic? And I made the observation, if you look at things in life, many things are arranged in pairs. So why not let's do it as a 2-chain? So that was my approach to starting out with a 2-chain model when he wanted to continue working on a 3-chain model. We'd actually failed numerous times with the 3-chain approach.
Rick, any reflections on that?
I would agree that, what we did as far as the structure and identifying the structure of DNA, there was a lot of insight in that, but the structure was one that wanted to be found out, if you will. It explained itself once you understood enough about how things went together. So we were amazed, again, at Jim's point of view that, using the 2 chains, of how easy the pieces fit once we agreed on the mechanism that we were going to use in putting our small pieces of metal together. And once we started to construct it, it made itself. So we were just it was just passing through us into this model. And you could do nothing but just stand back and say, that's it.
We would like to take this opportunity now. We have a phone back at our mic, and we would like to invite any of you in the audience, if you'd like to call this radio station, we would be more than glad to field your questions. You can direct them to Dr. Oppenheimer, to Dr. Watson, or to Dr. Crick. That works fine, and there's a mic.
Thank you. This question is directed.
Just about to say, this she's very famous, and this is Dr. Rhena. She has a big program. Oh, Dr. Rhena.
Thank you. Yes, Dr. Rhena, I have a question directed for both Dr. Crick and Dr. Watson.
Okay.
Gentlemen, what are your thoughts on genetic manipulation in relation to altering human behavior?
Well, there is a road that is taken to wisdom, and on that road you have to pass through knowledge. And I think what we have now is we have more knowledge about how to do these, but I'm not sure that we've reached that point where we have the wisdom to take the steps to work with the manipulating of the genetic material, inserting and turning on these particular genes when we see fit versus when nature calls for it. So I think we have a ways to go before we're ready to make a commitment to doing such things.
Wouldn't that be a natural progression?
It probably would be. I think at this point where we're on this threshold of completing the human genome map, we have many years in understanding that, what's there, what is, and how much of behavior is actually influenced by genetics and stuff. But one of the things that we have to look at from a perspective in our societies, and this is not popular, and I've been criticized for this, especially in the Human Genome Project. In science, we tend to push forward, especially with empirical science, and that's where we were working from, is trying to find out what this DNA stuff was, how it was put together. And then from that empirical work comes applied science. So how can you utilize that information and come out about with new technologies and such where we could do gene therapies or potentially alter someone's behavior with some of these therapies that might come about. But ultimately, when we're dealing with those levels of questions, that's a societal thing. And that's where we oftentimes get ourselves in trouble from a scientific perspective. People look at us and consider us to be cold, uncaring. I was considered to be the infant terrible of molecular biology for my work on the Human Genome Project. My main goal was pushing forward and solving this information problem. But it's unfortunately, it's society's question to answer. If we didn't describe this stuff, if other people didn't describe this stuff, others would have come along and done the same thing. And that's sort of an unfortunate situation we find ourselves in science, but it's the way it works.
At this point in time, within the scientific community, is there any safeguard to prevent manipulation that I've been describing, or you've been describing?
Well, and you could potentially have safeguards. We have safeguards that are in place as far as what people can do for transgenic organisms and how those might be potentially released into the environment and such. Part of the problem is, as Dr. Oppenheimer pointed out, is this whole business of rogue nations and such. We can, as a society in the United States, decide what we want to do with this information, or other industrialized nations. But if this information does happen to come and the technology comes into place with other countries that have all the gain and nothing to lose, that's a totally different situation. And I think the whole issue on the cloning aspect of organisms illustrated that concept, because there's several people stepped forth and said if it's banned in the United States, I'll take it elsewhere.
Another thing that I find interesting about this particular question is inherent in that question. We know that DNA is our genetic code. We know that DNA is going to code for our proteins. So trying to find out what the DNA code is, trying to find out what proteins can be turned on or turned off in their synthesis there is a very fascinating question. Underlining that in that particular question was, we're not really sure at this point in time, is all of behavior protein-based? So that is something that's out there on the horizon that will be very fascinating to find out, because there may be things in behavior that are not protein-based, and everything we know about the genetic code at this point is associated directly with proteins. So to make that quantum leap, if I can use that word, to saying that behavior is protein-based is quite a fascinating question, and we very much appreciate you for bringing that to our attention. Do we have further questions from the audience?
Thank you for addressing my question.
Thanks.
This question is addressed to Dr. Watson and Curt. I've read that a number of authorities believe that Rosalind Franklin should have been a co-recipient of the Nobel Prize that you two were awarded. Rather controversial. I'm wondering what your response to this opinion is.
Well, I think unfortunately Rosalind didn't get the credit that she so much deserved. She was working under Wilkins at the time as an assistant, but again there was a bad relationship between those two. And one of the things that happened is she gave us some keen insight, not only through her X-ray diffraction pictures of DNA, but also some other criticisms about our modeling, or at least our early modeling approach, that really directed us down the right path. But unfortunately, she died of cancer. And what happens is the Nobel Prize is not given posthumously. And we would have definitely expected that she would have been the recipient rather than Wilkins. Because the Nobel Prize, people oftentimes forget about that, but it was actually awarded to Wilkins, Watson, myself, and Crick. So there's actually 3 of us that were awarded that. And Wilkins more than likely would have not gotten that prize. It would have more likely been Rosalind if she would have still been alive.
Excellent, thanks. Other questions from the audience?
I have a question for Oppenheimer. I have a question both for Oppenheimer and for Watson and Crick. And I'll start with Oppenheimer. What do you think would have been different about fields of physics and chemistry if they're in this century, if they've never been in World War II?
Well, an interesting question. In some regards, the war set back physics because men like Leo Szilard and Enrico Fermi and others who would have done work in pure physics were either chased out of their native countries or left as refugees, or found themselves giving their enormous mental capacities to the creation of a weapon of mass destruction, which compromised them both philosophically and spiritually. And most of the scientists who were engaged in the Manhattan Project and other projects relating to the war, in some sense, never re-entered the world of civilian physics. Their lives were permanently distorted by world events. And furthermore, I said in my remarks this morning, after the war, I concluded that physicists have now known sin. This is a paradox that's built into the heart of science, and I'm guessing that was the tenor of the previous question. For a long time, scientists pretended that they were value neutral, and they were allowed, in some sense, to maintain that illusion. But in the 20th century, one of the dark implications of world events and the triumph of the scientific revolution is that scientists are now politicians, but politicians are not scientists. No politician understood the bomb, but we understood the political implications of what we were doing, and therefore we had to take charge politically in a way that we are not trained for and did not seek when we gave ourselves, in a sense, to the Hippocratic oath of pure science. And so we have been imposed upon, thanks to the triumph of the scientific and industrial revolutions, with a burden to help society sort out questions that are, in fact, beyond our capacity and which we don't deserve, because we are not representative of the mass of people who must make those decisions. And so there is a problem that's going to get only worse in the next century, that scientists know far too much and politicians know far too little. And those decisions are going to need to be made, and when politicians decide which gene to manipulate to prevent mental retardation, they're going to have no choice but to turn to you, and you're going to have to make decisions which are beyond the capacity of the scientific temperament, and we face that. So if there had been no war, life might have been much more complicated. The age of innocence for science might have lasted a bit longer. But unfortunately, the war came, and stopping Hitler was almost to every physicist's mind the supremely important act of his life.
For Watson and Crick, I want to ask, what do each of you think would be the worst thing that genetic science could be put to use for?
I would have to say it's not being careful with what we're trying to do. Some examples of what could come of this is when you mean to do something positive and it ends up being negative. There's been discussion on if you could, through gene therapy, get microorganisms that could take care of our petroleum pollution problems when we have large oil spills or need to clean up areas, that we could just introduce a bacteria, for example, that would use that as its food source and take out those petroleum products from the environment, which would be a very positive thing. Take into consideration then what would happen if they got introduced into our world oil supply. That we have to have that foresight of, yes, we're doing something positive, but is it worth the risk of what could happen next? It usually gets back in discussion to the human level and what sorts of manipulations are we willing to accept. If I say that we could cure disease A by doing this and everybody is in favor of that, well, does that lead to the next step of making people stronger or faster or smarter or musical? Will we allow that? And, again, like Jim said, keeping it contained in the United States will not be as difficult as keeping it contained when this ability spreads throughout the world, because our wants aren't always another nation's wants.
I think one thing that we have to kind of be careful of too is that we don't repeat the pattern of this last century, because one of the things that we could do, especially with this business of genes here, we could probably all agree that it would be appropriate for us to do gene therapy to get rid of various types of diseases like cystic fibrosis, these types of things. But once it gets into this arena that maybe if we do find that there's a protein link to behavior, those type of things, where do you decide? What's the deciding line on that? Where you're going to get rid of genetic diseases, which we could all agree on, and then when are we going to switch over to, well, maybe we'd like to have our society or people in our society behave in a certain way. So we're getting back to potentially that slippery slope of eugenics, negative versus positive eugenics that Hitler was involved in. So we might be putting forth a notion that we might have to reconsider those kinds of questions. I don't think the answers are real clear. Thank you.
Thank you very much. And we have another question.
Dr. May, I would like to ask this question of Dr. Watson and Dr. Curt. This morning, Dr. Oppenheimer said that having witnessed the detonation of the bomb, that he had become deaf. And I wonder if, after you witnessed your creation of what the DNA molecule looked like, if you had come closer to witnessing what it's like, do you think that your discoveries have led us closer in science to understand just the origin of life?
That's a good question. Things that, because I left a few years after we did this research, I have just kept up with it like most the scientific community, reading about it. But it is very interesting to see the ties that by understanding DNA and the products of it and RNA and making comparisons between what has taken place in what we call the kingdoms in science. And when we look into the human DNA, we recognize structures of DNA and RNA that are more bacterial in origin and eubacteria. And so there has been, it is hypothesized, a mixing of DNA across these kingdom lines, so to speak. And so I can't speak directly to the origin of life, but the complexity of how a cell has come about and become specific, I think has been enlightened by understanding DNA and RNA.
Yeah, and I think one of the things that you could look at there is when you figure out that information on sequencing, and this poses a lot of work. We're working on this human genome map. We're almost done with that. And this has been proposed, this gene sequencing, where you map the entire genome for a number of other organisms, Drosophila, the fruit fly, and several other critters that we use frequently in a lab situation. We could potentially answer those questions, but it'd be quite an exhaustive project, getting enough pieces of the puzzle to sort of fit that together. So it's another one of those things down the road. So when he had this concern with his mother that there's not going to be enough for me to do in science, we've opened the box here, so to speak, as far as the amount of research it could be potentially done concerning this.
I think when I busted into the Eagle Pub and announced that we had discovered the secret of life, I overstated myself a bit.
So my queasiness was warranted.
Thank you, General. Do we have further questions?
Dr. Renee, thanks so much for taking my call.
Yes.
I have a question for Dr. Crick, and my question is this, Dr. Crick, how did you become interested in brain research after your work on DNA? And the second part of that is, has your research now linked, because I would assume when you're doing brain research, you're really interested in behavior. Have you been researching the link between, say, protein-based behavior and your brain research?
The answer to the first question is before I even attended Cavendish, that my two interests were, and neither one had a title at the time. There was no such thing as molecular biology or neurobiology, but I had an interest in both, and I kept that interest in neurobiology all the way through my work at Cavendish. So when that project was finished, I just decided to move into the neurobiology. The neurobiology work that I've been doing has been more with how do neurons in your brain constantly reorganize themselves. And the computer system has come into play because it is a system that we've organized, and we've considered that that's probably the way the brain works, but we have found out through our models, because most of what I do is just theoretical, I don't do any experimental work, that that's not how the brain works. The neurons don't line up in the end. They seem to stack and lean over each other. So when something new comes into our brain, we have to go through the whole deck of cards and reshuffle how it all fits together. So I haven't been working in the DNA end of the neurobiology. It's been more when I see something, how does my brain process it and know I'm seeing what is being projected in my neurons, and how do I know that I'm conscious and aware of my environment. Those are more the problems I'm looking at.
So do you think there's a model, as you're talking about the stacking of the neurons, that might have a link to the DNA model?
Well, what we have done is we've taken our research offline, so to speak. We've taken away the computer's ability to reorganize and moved it offline and just said, what would you do with this thought? With multiple stacks of neurons on each other, and push the offline button and see how it shuffles, how many different ways it can make connections between these thoughts, which has led us into the belief that that's possibly how dreams work, and that we have our processes that we have realized through that day, this day, and our brain, as we go to sleep and hit that REM sleep, it's going to shuffle those cards again and look for connections, no matter how weird they may seem when you're dreaming. Where do I plug these in? What part of my whole thinking system does this become?
Thank you.
Excellent. And we are still here live at KNIC and waiting for your phone calls. Ring.
Hello, Dr. Renee. I have a question for Watson and Crick. Speaking of cards and puzzles, I'm dying to know how they figured out that half of the double helix, the nucleotides and the sugar and phosphate backbone, how did you figure out that it was upside down?
Would you care to remember?
Are you avoiding it? When you have the hydrogen bonds, by Donahue to pay special attention to the tautomeric forms of the bases, which means that they can move in between two forms, a ketone and enol form. And when we stuck with one form of that, those bases, they lined up upside down. They wouldn't line up when you paired them with the 3' towards 5' running in the same direction. And only by turning those over would they actually connect and the whole model would follow. So that's how it ended up, one side ended up being what we would call upside down.
Excellent. Does that answer your question?
Yes, it does.
Great. Any other phone calls to come into KNIC radio station this evening? 3.
Yes, I want to thank you for taking my call. I'm going to go ahead and put this on speakerphone and I'll just talk to that.
Okay.
For Dr. Watson and Dr. Crick, how do you respond professionally to the advancements in cloning, the incident that we had recently with the sheep?
One thing I think we have to take into account is both of us having some background in biology. We have to realize that if you look at our bodies, we are one huge mosaic of clones. One of the things that takes place is we start out as a single cell and our cells are dividing. All those cells are genetic clones of one another. And what's happened then is we differentiate and develop in our tissues. Certain genes are turned on, other genes are turned off, so we end up producing the right kind of cell type in the right location. So when you look at this process of cloning, and basically what we're doing is we're taking a cell, we're knocking the nucleus out of it, putting a foreign nucleus into it, and getting the whole process to start over again. We're taking something that we already do to a certain degree. And we like to point out here, our radio announcer here, she has her own set of clones. She has identical twins. So the whole process of cloning, you hear a lot of people making sort of a big statement about the fact that we have the ability to do this now. But humans have been cloning when we look at this business of identical twins, and then we look at our body. All of our cells are genetically the same. It's just there's different expressions of those genes in the different cell types and the tissues we find in our body.
My perspective on cloning has always been pretty interesting. One day, I have 15-year-old daughters that are identical twins, and one day somebody at the house at a sleepover was talking about, oh, cloning this or that, and they just looked at each other and went, yeah, what's the big deal? We don't have a clone, they live right in the same house with us.
I think another thing that may come from this cloning, other than whether we can raise a strain of sheep or a strain of cattle that are more productive or produce what we want, is that one thing that is yet to be figured out is how our cells, spatially and temporally, different. If all the billions of cells in our bodies are genetically the same, why did the cells in my hand become a hand and those in my foot become a foot? And why do certain things happen at certain points in our life? Why do you grow hair of a certain color up to a certain age and then either it continues or it stops, or it changes color, or it doesn't change color, it becomes curly, it becomes straighter. And through cloning and understanding what takes place when you move that nucleus, that genetic material into another cell, and paying close attention to these embryological stages, we may be able to figure that part out.
Many biologists think that's one of the real key puzzles yet to solve is developmental biology. How do we go from a single cell at fertilization until you form this juvenile form, then that grows into an adult? How does all of that patterning take place? That's a puzzle in biology that still, many questions have been answered, but many questions remain.
Excellent. Thank you, Watson and Crick. And do we have anyone else who would like to speak on the speakerphone?
Yes, speakerphone. Dr. Rhena, I have a question for Watson and Curt.
Okay.
It has been said that science should be amoral or without morals. So is this saying in your perspective that as scientists you should not choose what happens to your research, you should simply do it, or that science should have a choice in where research goes and society should not?
Excellent question. Gentlemen?
It's a very complicated issue. There's many factors in that. Where you work depends on how free your information is to society. A lot of times if you're working in a funded lab, what you produce is the lab's, it's not yours. They make that decision. I think it needs to be understood that we live in the same world that everybody else does, and even though we may use words like amoral and disconnected, we participate out in society, and we have our opinions on how things should take place. So we don't want to see bad things happen either. It's not that we want to just stand back and say, here it is, you do with it what you want. Because when society does it, it also happens to us and our families. So I guess the answer is no. In short, we'd like to see beneficial things occur with the research that's come out.
I think the amoral aspect is more from a focus of how science works, where we want science to work in the process of being unbiased. So we're doing good, high-quality, objective kinds of work. There was a case not too long ago out of MIT where an investigator was doing research, and there was actually a subordinate member that was making up data for a research project that she wanted to see grant funding continue, and another subordinate underneath this person blew the whistle And what happened was they ended up trying to get rid of the whistleblower. So when we talk about science being amoral, the main thing is we want to let the data speak for itself and let science advance in as unbiased manner as we possibly can. And like Francis mentioned here, we have families and we're concerned about some of the spin-offs that come out of this. But that's a societal thing. That's outside of the lab environment itself. So when we talk about science being amoral, that's from the lab perspective, when we're doing pure science, trying to figure out the way the world is around us.
So do you believe that society should have no say in what becomes of things such as genetic material, DNA, or should that be something that is partial society, scientist choice?
I do think it's a scientist's job to educate on what the implications are. Just like there's a gap between scientists and politicians, there's becoming a larger gap between what you're referring to as society versus the scientists. And it becomes very complicated to try and keep up. So before scientists propose something, they should be sure that the public is educated enough to understand what they're suggesting. And only by doing that then can we ensure that there's a fair vote on how it should be treated.
And we have some more call-ins.
Yes, Dr. Renee, as a follow-up question, I would like to address this question to Dr. Oppenheimer, who's had considerable more experience, as my question is going to indicate. Earlier, I heard Dr. Oppenheimer this morning speak of we can't put the genie back in the bottle, but at least we can put the toe of the genie in the bottle. Is there anything that Dr. Oppenheimer could use or could address to Dr. Crick and Watson about the genie that's now out of their bottle?
Well, again, there's a very significant difference between what I did and what these gentlemen have done. My project was conducted under the greatest code of secrecy in the history of science. And in fact, that proved to be a very difficult problem in recruiting the ablest physicists to work on the project, because it's counter to the very spirit of science to engage in closed discovery. And several of the scientists argued that they could not possibly agree to the strictures of the United States Army in participating in the Manhattan Project, that it somehow was a detour from centuries of scientific progress, that we should engage in something that would be husbanded for the benefit of one society, not for the benefit of all. And I think that was a setback in the history of the freedom of scientific exchange. One of the breakthroughs came when I was able to persuade Leslie Groves to create a lab at Los Alamos. His plan, the Army's plan, was to scatter all of the participating scientists around the country and keep each of them in the dark about what every other one was doing. And somehow the results of all this would be coordinated by a tiny cadre of people. I realized that not only is that counter to the traditions of science, going all the way back to Bacon and beyond, but furthermore, it is not a good creative process. I think one of your questions earlier was how you create true collaboration. And true collaboration involves one scientist calling another's bluff or challenging another scientist in just the way that leads to the next breakthrough. And so I demanded, as a condition of my own employment in the Manhattan Project, that we create a site where there would be relatively open discussions. This drove Leslie Groves nearly insane. But in Los Alamos, we did create an enclave, at least, where such discussions were encouraged. We had strategy sessions once a week in which anything was allowed to be discussed. Now contrast that with what you have done. There was a small amount of proprietariness that you've each admitted in your own work, trying to get in first, trying to defeat in your discovery arrival. But that's actually, I think, a healthy instinct in science, so long as it doesn't go too far. But your work has been given to the world, and any person who cares to can understand it in its entirety. No part of it is kept in a locked vault. And so I think the differences between the implications of what we did and the implications of what you have done are enormous, and particularly for the Cold War. The Cold War, in some regards, if you take Leo Szilard's philosophy, the Cold War was made inevitable by the fact that the United States was so secretive about its own atomic research that the Soviet Union rightly resented the fact that our major ally in the war was kept in the dark about these secrets and therefore overreacted to what it took to be an atomic gap and created an arms race that might have been at least attenuated and possibly even eliminated by a more open exchange of ideas. So I congratulate you on the openness, which is true to the traditions of science, and decry the closed secrecy that was unfortunately inevitable, perhaps, in my own project.
Thank you. And we have another call.
Dr. Rhena, as your executive producer, you know you have to limit the time on radio. From 7 to 8:30, your time is almost up, and I know that your viewers would like to hear our very distinguished guest, out of character. And let me say, one of your guests tonight will be at 10 o'clock tomorrow presenting in the Sheila Auditorium. He will do a great keynote speech on the 20th century. And then Mr. Kim Riley will be here from Boston to do the Beatles at 11 o'clock. So I'm sure your listeners, they're not viewers, you're not used to television. They will want to come to that. So at this time, could we have a character and then we can adjourn by 8:30?
Excellent. Okay, Dr. Watson.
Well, my name's David Foster, and I teach biology here at NIC. One thing that I find exciting about all the stuff with DNA, my interest is in natural resource management, and I teach several courses here at the college on wildlife management and rangeland management. And we're kind of coming of age in my area. We tend to be sort of a bit behind the times. We're relatively young sciences. Wildlife management you can trace really to the 1930s, although Leopold was sort of the founder of that. Range management to the late 1800s, Arthur Sampson is considered the father of rangeland management. But we're starting to come of age with DNA. One of the things that's happening now in the state of California, poachers beware, we're starting to use DNA fingerprints when we find gut piles. So when we find a gut pile, we can identify it from the meat that's in your freezer, from the trophy head that's on your wall. So we're using these high-tech things now to go after people. Also in the area of wildlife research, they're doing studies in the grizzly bears in Yellowstone National Park. It's kind of interesting. They'll use basically carrions set up with a triangle of barbed wire fence surrounding the grizzly bear, or the carcass, that they have in the area. Grizzlies will come in and deposit hair on that barbed wire fence, and then what they're doing is they're taking and analyzing the DNA of a little subcellular organelle called the mitochondria. And they're delineating a lot of information about the bear population, how they're related to bears in Alaska, other continental grizzlies up in Glacier Park, and so forth. So we're learning a lot of information from the biodiversity perspective. We're also seeing it from sort of an applied area as well. So there's many exciting developments that I'm seeing in resource management coming out of DNA, and I would have never envisioned something like this when I went through my training in the early 1980s. No one was talking about DNA applied to that area, and I'm sure we're going to see more of that expanding in the future.
Thank you, David. And Dr. Curt?
I'm Mike Curt. I also teach biology here at North Idaho College. And more on the philosophical point of DNA, I find it fascinating that the DNA that's in all life currently has survived because of the way a cell replicates itself. So the codes that we carry around in our body and the plants carry around and the protista organisms and the fungus and the bacteria have survived all of the plagues and all of the world events that have caused destruction, and we represent those survivors in a sense. What's going to allow us to overcome many of the maladies that happen in the future is that we carry this blueprint inside of us that says, here's how you overcome it. And once this project is finished and as it carries on and figuring out what genes do what and what protein is produced, we'll have those answers. It's a huge library that we're just trying to decipher, and it will take us through, I think, a lot of the problems that we're going to run into.
And Dr. Oppenheimer?
Oh, I don't know what to say. I've really enjoyed this. My goodness, everything I now know about DNA I've learned in the last hour. I'm interested in the implications of all of this because let me give you a couple of quick examples. But I've been going back over and over again to Oppenheimer's statement, "I am become death, the shatterer of worlds." Now, that's a very wild thing for him to have said. He actually produced a fair amount of resentment in the scientific community for saying that. And you can see why, because there's a certain kind of righteousness about this that really clouds the very issue that we've been trying to get at, which is where does the scientist end and the culture begin, and where does the scientist end and the politician begin? What are the ethics of science, and so on. So I think there are very severe implications for this. I have, in my life, a mentally retarded aunt who had Down syndrome. And she died 3 or 4 years ago at the age of 52. When I think of my own life, what shaped me, what gave me values, what taught me tolerance, what added perspective to my world, I can't imagine Susan not having been in this equation, and yet she won't be in this equation. She will be genetically repaired in utero, if it even gets that far. And 15 years from now, doctors will just routinely say, I urge you either to go in and repair this or not to permit this child to come to term. Now, I can understand that for spinal bifida, and I can understand that for profound genetic failures in utero, but what about things that aren't necessarily profound? What about shortness? What about lack of athletic ability? What about this and that? So this raises some very significant issues. Use the metaphor earlier, shuffling cards and sleep patterns. The pattern of life up until very, very recently was that life is a hand that's shuffled by God, and you make the best of whatever cards you've been dealt with, and the community absorbs the disabled in one form or another. Well, we're reaching the time when blissfully we may be able to rise above some of that, but the implications of it for the mix of society and the perspective of the society are profound, I think. And of course, I'm giving you worst case examples, but what about the person who wants to micromanage her child so that it's a chess player rather than a football player? This is probably within the realm of possibility within the foreseeable future. So that seems to me that that Oppenheimer was right, that at the end of the 20th, in the middle and the end of the 20th century, we have become so profoundly committed to the scientific paradigm that certain things are going to play themselves out whether we like it or not, and the politicians and the constitutions just can't possibly keep up. And, when he said, I am become death, it's possible to read that in a whole range of ways, but one of the ways it's possible to read that is a certain kind of fascination, a certain kind of death love that this man had, and that maybe science has, or maybe civilization has. I'll give you a quick comic story. When I was in high school, I lived in western North Dakota, and I got a chance to go work on the only ABM missile site in American history, in Langdon, North Dakota. That in itself was a sort of death obsession, I guess, but I went up there for the summer and lived in a trailer, and one way we eked out our limited little income was that we had a friend of mine, and he was much older than I, but we had a fireworks stand. And he left one day. This is kind of Sorcerer's Apprentice story, unfortunately, but he left and left me in charge of the fireworks stand, and I sold this and I sold that, and then I got it into my mind as I was sitting there bored on a June afternoon in North Dakota. Here's the following question. I said, how close? I can't even imagine how stupid this is. I thought, how close could I put a match to the fuse on this cone and not light it? Now, there's only one way this story can end, of course. And so eventually, of course, the fuse lit, and I panicked and threw the cone out in front, and it blew back at me, and 5 or 10 other things started to go up. And I wasn't killed. I wanted to be killed because I would have to face my business partner. But this was one. I mean, this shows either A, that I'm just a complete idiot, or B, that there's something in humanity, possibly just maleness, which just likes to play with the fuse. And Robert Oppenheimer liked to play with the fuse, and he did. And when he saw it, he realized he was as stupid as I was. But there's something possibly right at the center of our genetic instruction or our coding that makes us brinksmen. And Arthur Kessler went so far as to say there was a bad gene in humanity. And I recommended a book last year, so I wrote it. Rebuked me for it, but I'll recommend a book this year. I mean, it seems arguably the most important book of the 20th century is Sigmund Freud's Civilization and Its Discontents. Very short book and a profound one. And Freud basically says that humankind is a being bent on chaos. And it takes every bit of every civilization working day and night with all the repressions and religious codes and regulations and educational authority to keep people minimally in line. But then he says in the middle of this book, what's happened since the scientific revolution of the 16th and early 17th centuries is that man has become, and this is his great phrase, man has become a prosthetic god. He has a prosthetic ability to manipulate matter, so a crane is a super hand, and a telescope is a super eye, and a bomb is a super fist, and so on. So here we are at the end of the 20th century, early 21st century, and we become a prosthetic god of almost unbelievable magnitude. And here we are again at the frontier of another one. And what will we do with it? Freud's view is, you're the prosthetic god, but you're the same old sick person manipulating the prosthesis that you've always been. And so this is a big paradox built into the nature of Western civilization, I think. And I think Oppenheimer was in love with death. His favorite poet was John Donne. John Donne is the poet of death. And when he read the Upanishads, he didn't read stories of victory and creation, he read stories of death. And so it's in a sense not surprising that it was Oppenheimer who was thrown up for this task. It seems to me that this is a big paradox of science and civilization.
This is Rhena Cooper with NIC Radio, and as a microbiology teacher here at NIC, I just want to thank you so much for coming this evening and talking, and I hope you keep talking, I hope you keep asking questions, I hope you keep mulling these things over in your life. It's just fascinating that we're to the point where we can look at things and we can be open-minded and we can try to deal with these issues, because dealing with these issues is what's going to enable us to survive the next century. Thank you, and have a wonderful evening.